everyone, and welcome to StrandExplains. As always, I'm Sakshi, and today I have the good fortune to be joined by Dr. Harsha Gowda. Hi, Dr. Harsha, how are you?
I'm good, thank you, Sakshi. Thanks for inviting me on your podcast.
Of course, thank you so much for joining us. So, Dr. Harsha had did his bachelor's and master's from the University of Mysore, followed by his PhD from the Institute of Bioinformatics in India and Johns Hopkins University in America. He currently is working at the Queensland Institute of Medical Research, researching cancer biomarkers and their targets. So today we thought we'd ask Dr. Harsha to talk about cancer proteomics and how they and just understand what they are and how they work and why research in them is really important. So I thought maybe to start off, Dr. Harsha, we could you could talk about what led you to study cancer omics in general.
Yeah, so cancer is one of the most prevalent diseases, along with cardiovascular diseases in the world. And if you observe, as we improve average life expectancy throughout the world, you expect to see a higher incidence of cancers because predominantly it's an old age disease. Although there are childhood cancers and there are some cancers that afflict the young, but predominantly most cancer patients, if you observe, will be relatively old. So that way, you could assume this will be a major health burden for almost all countries around the world, and it's an important problem to tackle. And healthcare wise, it's an important area for there is significant room for improvement too. So that's why cancer and why omics. Omics essentially allows you to investigate any given problem in an unbiased way and in a high throughput way. So that way, you don't necessarily go with one particular hypothesis, spend a lot of time testing that hypothesis. You could say, okay, if here is the problem and you have a certain sample size, let the data tell me more than me speculating and going one by one whether this hypothesis is correct or this hypothesis. So that's why I think most of the omics technologies kind of took over in the last couple of decades. And genomics is pretty much now omnipresent. And some of the other omics technologies are widely practiced, but may not be as prevalent for various reasons. Yeah.
I see. And yes, like you're saying, omics, genomics specifically, or genetic omics is very popular right now, and use a lot of the research you hear about, or the popular research, is typically genomic research. And I mean, even in our podcast, all of the most of the research we've spoken about so far has a genetic basis. But like you're mentioning, like you just mentioned, there are other omics. For instance, cancer proteomics, which is omics of the proteins, is slowly gaining traction, especially in cancer proteomics. So could you explain what exactly proteomics is and why it's important to study them, and maybe put more emphasis on that aspect of research?
Yeah. In very simple terms, proteomics is a study of proteins, basically, just like genomics is a study of genomes. So, and omics particularly simply because you can study proteins in, again, high throughput manner, a large number of them at a time. Why they are important? So if you look at any biological, a cell basically, so we say genome is the blueprint. That means in my body, every cell pretty much has the same genome. But what varies is what parts of the genome encodes set of proteins for specific functionality, identity of a given cell, or phenotype of that cell, or behavior of that cell. So that way, protein is dynamic. Like genome is somewhat static, that means all cells have the same blueprint, but protein expression is very dynamic. That means I can take a brain cell and it will have a set of proteins that are encoded by the same genome, which allows a nerve cell to do what it does. And you take a liver cell and again, there is a different bunch of proteins which are important for the functionality of a liver cell. So because of that, understanding proteins will give you a portrait, a biochemical portrait of any given cell or tissue type, how it operates and what are the players that regulate it. And if you look at it, almost all the drug targets that we use in the clinic are proteins. Like you're targeting any small molecule inhibitor or antibody or any form of drug against a specific protein to either block an activity or to, let us say, enhance a particular activity or supplement an activity. So also as biomarkers, like when people diagnose a disease, most of the markers that we use, biochemical markers in clinics, are also proteins with some small molecules. So that's why it's important to study proteins.
I see. Thank you so much for that. I apologize for the lighting change. Bangalore,
Sure.
power cuts being what they are. Thankfully, my internet is hooked up, but yeah, just had to deal with this.
Yeah.
Yeah. So you mentioned that while basically the genomic map is static, the proteomic map is more dynamic. Sorry again. But so
Yeah,
where
that's
would
fine.
transcriptomics fall in this? Because transcriptomics, which is the study of the RNA of the cell, would also be more dynamic in that it's the RNA is what makes the proteins. So why study proteomics over transcriptomics if they have similar, if they give you similar information?
Yeah, that's a good question. So RNA is like an intermediary between the genome and the proteome. So what you can definitely gain insights from studying transcriptome or mRNA expression. It can serve as a reasonable proxy for what proteins may be expressed in a given cell type or tissue type. But what it can't tell you is because proteins also undergo a lot of modifications. That way, for any given protein product of a given gene, there could be hundreds of variants, whether they are alternatively spliced variants or post-translationally modified variants. Like that's why when we say phosphorylated protein or acetylated protein or glycosylated protein or proteolytically cleaved product, for example, most of the hormones, including insulin, they are not doing their function as they were encoded by the genome. They undergo further processing inside the cell to create that functional product. So that way, transcriptome is a reasonable proxy for protein expression, although there are enough evidences to suggest how sometimes it does not necessarily reflect the protein levels. There are enough cases of that. It does not directly correlate. But what any of the other methods cannot reveal to you is how these proteins are active in a given cell, like what modifications are either inhibiting their activity or activating them, and what you want to target. So these aspects are not easy to predict based on just the genome or the transcriptome. That's why you have to study protein.
I see. So could you maybe say that studying proteomics gives you a kind of real-time snapshot of what's happening in the cell at that moment versus?
Yeah, and also much closer to the phenotype. In other words, you are when you go to genome, you are several degrees separation from the actual phenotype because it is there, and you may understand certain sequence variations and what they can do, but you are still at a distance and you don't know exactly how it manifests. But as you go closer and closer to proteins and some of the biochemical entities, you are much closer to what is ultimately influencing phenotype of a cell.
I see. Okay, that makes sense. Thank you. And so could you maybe explain how studying a cancer patient's proteome affects their treatment and what understanding their proteome means in terms of or in how it can inform their treatment?
So if you think about any disease, including cancer, so let us say under normal circumstances, there is a certain way all the genes coordinate, orchestrate, and carry out certain functions. Like if it is kidney, there are genes that are expressed and coordinate the functions that are supposed to be carried out by kidneys. And even when the cells are dividing, it's a highly regulated orchestration of many different genes to precisely generate enough amounts of different genes or proteins that are required for proliferation. So in cancers, in simple terms, it's just aberrant regulation or uncontrolled division of the cell because of the aberrant regulation of the genes that are supposed to be carrying out these normal functions, right? So from that point of view, if you study proteins and say, okay, what is different between a cancer tissue or cell compared to its normal counterpart, you get to directly understand which are those proteins that are differentially regulated between a cancer cell and a normal cell. And if these are the list of proteins that are differentially regulated, which of those could be targeted to essentially overcome or limit the proliferative capability of the cancer cell, which is what most of us want to do in the field of cancer. So that way, it's important to study proteins, and that's how it provides insights. And also, more and more, if you look at some of the literature in the last few years, we know that there are several signaling pathways, which are normal signaling pathways under normal circumstances, which are required for cell to function, whether to proliferate, whether to produce some hormone, or whether to sense something in the surrounding and react to it. There are various signaling pathways that regulate these processes in a cell. And in cancers, some of the proliferative signaling pathways, that means which signal to the cell that they have to keep dividing, are high, and some of the anti-proliferative signaling mechanisms that are supposed to keep it in check are dysregulated. It is like you have a car, and if you have access to both brake and the accelerator, you kind of manage how you want to go on the road. But imagine I just completely take away the brakes, and now you're at the mercy of the accelerator. So that's what's happening in cancer, essentially. There's no negative force blocking it. And that's why you study these processes through proteins so that you can target them.
I see. And I really like that metaphor. It's a great metaphor to help understand how these things work. I did have a question just about the limitations to proteomics, or why it might not have been studied as widely. But before I get to that, I thought I'd ask you, since you just mentioned, this model of the accelerator versus the brake, and how these signaling pathways sometimes go unchecked. Can you maybe talk about that in terms of checkpoint inhibitor based therapies and immunotherapies, and how those are sometimes used to deal with such problems of overproliferation or tumor suppression?
So yeah, good. So it's a segue, but let us connect to cancer. So what is essentially happening in case of checkpoint inhibitors, at least in cancer therapy context, is, see, even in body, we have all sorts of tissues made up of all sorts of cell types. And one of the fundamental ways you are protected from the onslaught of many other pathogens and microbes that are constantly looking to, let us say, invade or establish themselves is your immune system. So that way, there are two, when we think of immune system, whenever we study in textbooks, most often we end up focusing on a lot of these immune cells circulating in the blood, like which are part of the blood. But also we have to remember almost all tissues, whether my skin or whether it is intestine or whether any tissue in the body, there are several resident immune cells. That means in addition to circulation, there are several immune cells embedded in the tissues itself, so that locally, whenever there is something wrong, these immune cells can do what they are supposed to do to protect the host tissue. Without which you are waiting for, let us say, the immune cells in the blood to sense it, then infiltrate, then come and do the job. So that way it will not be a most efficient way. So that way you have both circulating immune cells in the blood as well as resident immune cells in different tissues. And there is reason to believe, yes, these immune cells also, the blood immune cells also infiltrate, and some of the resident T cells may also get into circulation. Now, throughout our life, it's quite possible that, because cancer cells are an accident, like if you look at the mechanism of what really happens and why cancer cells emerge, you have each time a cell divides, there is a certain error rate associated with the DNA copying, right? Like it is something like, if I ask you to copy a book 10 times, I'm sure each time you copy, you will inadvertently make some errors, some spelling mistakes in some words randomly. It is not intentional, it is just the nature of it, basically. For every 1,000 words you wrote, maybe there will be one or two errors, let us say. Similarly, each time a cell divides, there are errors that get accumulated in the genome. Most of these errors are insignificant, let us say, non-consequential. You will not even realize, and the cell doesn't have any impact. But occasionally, if these errors end up happening in regions that are important for regulating proliferation rates or any of those, that's when one of the consequences will be this uncontrolled proliferation, because now you have an error either inactivating some of the brakes that were supposed to keep it in check, or activating the accelerator, which overdoes what it is supposed to do. So when such a thing happens, your immune cells also are supposed to sense something is off, and sometimes they will destroy those rogue cells without you realizing it. So that way it is quite possible throughout our life, when we are immune competent, cancer cells may emerge occasionally purely as an accident, and your immune cells may simply clear them because they recognize something is not right. Because your immune cells are equipped to recognize what is self, like what is our own tissue, which is not supposed to destroy, and what is foreign. And because cancer cells will have these changes, some of the changes that it accumulates, which makes it foreign, because it doesn't look like the regular normal cells, immune cells end up recognizing some of these and saying that, okay, something is wrong here, and it will destroy it. But one of two things happens. When the tumor emerges, some cells have this capability to, or they end up secreting certain factors, which is immune suppressive. That means which debilitates the ability of immune cells to recognize that, okay, here is a rogue cell and it is supposed to destroy. That means it will put a brake on its ability to sense things. Now, when that happens, that means your immune system or immune cells, which naturally can sense and destroy it, are now defunct. Like they are there, but they can't see. So it is like it is complete darkness for them. So in such situation, and people figure this out basically, like the immunologists when studying, okay, what are the, because you have to understand, even immune cells, people wanted to understand what stops them from attacking our own tissues, or what are the checkpoints, like how is it regulated? How does it sense that now it is supposed to act, now it is not supposed to act? While studying that, they realize there are these checkpoints that keep it in check, so that you don't have autoimmune disorders. That means it doesn't attack your own tissue. So one of those checkpoints happens to be a ligand called PDL ligand, programmed cell death ligand. And there is a receptor, PDL receptor on the cells, basically. So if this binds, it inhibits the ability of T cells particularly to sense and destroy or inhibitory signal, because of which now immune cells can't attack the tumor. So now the therapeutic intervention was, once people realize this, if I block that inhibitory signal, will that allow our immune cells to now do what they are naturally going to do anyway, if this inhibitory signal was not there? So that's why these are checkpoint inhibitors. So that means PDL1, some companies are targeting the ligand itself. Some people are targeting the receptor, so that the ligand doesn't, there is no space for the ligand to come and bind and inhibit the T cells. So that is in a nutshell what we are exploiting as far as immunotherapy is concerned, checkpoint immunotherapy is concerned. And that has been fantastic, because at least some of the cancers that were not easily treatable, and particularly those which have high mutation burden and high antigen load, they seem to respond extremely well for checkpoint inhibitors. Particularly melanoma and lung cancers respond pretty well. So that is in a nutshell cancer immunotherapy as it is available today. One aspect of it. There are other CAR T therapies and others that will, yeah.
Thank you. And I think that maybe going back to my previous question, I thought it was a great segue back then. Jumping back to proteomic studies, could you talk about what the limitations to proteomics are? So one thing we spoke about is, while genomics has had such a it has been so popular, and a lot of research has been done in genomics, unfortunately the same is not true for proteomics. And So could you talk about why that's true and why it hasn't been studied as widely so far?
Yeah, so there are a couple of reasons for that. Like, unlike a genome and a transcriptome, let us say DNA and RNA, you can't amplify protein. Like, one of the good things that that we can do with DNA and RNA is that you can easily design primers or use random primers, and you can amplify. That means even if you have the lowest amounts of the starting material, you can amplify it, generate more copies of it, and study it, basically. Unfortunately, that's not possible for proteins. You are dealing with somewhat absolute amounts. So that is one impediment. And then people figured out, okay, we can use mass spectrometry to study proteins. But even there, again, amounts play an important role, like you can't amplify. Um, the reason it is not as widely used, I suppose, is because one is lack of access in many places. Like, the technology is a bit more challenging, and the capital investment for some of these equipment is high, and the trained personnel with this expertise is also limited. And there are, because of lack of amplification, there are issues with what you can and cannot study and how much of starting material you need. So a combination of all of these has kind of rendered it not as widely practiced as genomics and transcriptomics. For example, there are so many service providers for uh, genomics and transcriptomics. Like, you could simply isolate DNA and RNA, just send it out to a service provider. They can sequence it, generate the data, send it back to you. So although there are service providers for proteomics, but they are not as widely available, as well as sometimes they can be cost prohibitive. So all those reasons has limited its penetrance, let us say, in the number of labs that routinely use it. But slowly, I can see that, okay, more and more organizations, institutions, and labs now use proteomics, because there are certain questions which you can only tackle using proteomics, because genomics and transcriptomics cannot address some of those questions. So there, it's inevitable that you use it. But slowly, if it becomes much more accessible, much more simpler, much more not so cost prohibitive, I think it will become much more widely used.
I see. That makes sense. And I'm I just wanted to point out that you're talking to a biologist when they use a word like penetrance in terms of market availability,
Yeah, Jen,
because it is a genetic term in terms of penetrance of a phenotype.
absolutely.
Yeah,
Yes, yes.
so that's how you're talking to a biologist. Um, so, one thing that I've just started reading about, and I was hoping you could talk about, is cancer neoantigens, which come out of proteomics. And if you could explain what they are and why they are also being researched in the context of cancer treatment as well as cancer detection.
Yeah. So, as I was alluding to just in the previous question about how immune therapies or checkpoint inhibitors work, and now just to tell you, okay, how do the T cells or immune cells recognize this is a cancer cell or a rogue cell or an infected cell? Because even when we have infections, this is exactly what happens. So essentially, all of our cells have a mechanism where part of the proteins that are produced inside the cell are processed, cleaved, and presented on the cell surface by a complex called MHC complex, major histocompatibility complex. So these, you can consider them like antigens, like they are presented on the cell surface, and your immune cells can sense these antigens. They can keep, that means this is a surveillance mechanism. Even to recognize it as self, if they see the familiar antigens that are coming from the host, the immune cells know that it is a native tissue, native antigens, so it doesn't do anything. But at the same time, if there is an infection, let us say, and there is a foreign protein being processed and presented by a pathogen, a bacteria or a virus, now if your immune cells can sense that, okay, there is this unusual antigen, and then it knows that, okay, that cell has to be attacked. Even in cancers, in most cancers, you do see accumulation of somatic mutations, right? So as the cell divides, all these errors get accumulated in the coding region, and some of these errors get incorporated into the proteins that get made from these genes. And some of these stretches end up getting presented on the cancer cell surface. So now if it is presented, your immune cells can potentially recognize them as antigens, because this is not the sequence that it was supposed to be. It has altered now. So now these are what people refer to classically now as cancer neoantigens. That means these are variants, somatic variants, coding variants that get presented on the cell surface and are recognized as antigens by immune cells. So the advantage of knowing them is, one is, if you know these are the neoantigens that are being presented on the cell surface, you could engineer T cells which can recognize those specific antigens, and that will become a therapeutic intervention in itself. That means now your cancer cells are presenting this antigen, and you are infusing T cells in large numbers that are designed to recognize and target cells that present these T cells. That's a way of therapy. They can also be leveraged as vaccines, cancer vaccines, right? Because just like we are going through the COVID, and what are we doing for vaccine? We are essentially trying to prime our immune system by artificially giving the antigen from COVID, so that your immune system is primed, so that when the real pathogen comes in, so your immune system already is geared to attack it. Similarly, if you have cancer neoantigens, you could potentially prime your immune system to recognize it, so that once they emerge, they can attack it. But the problem is, most often, especially for a company like Strand, which does a lot of cancer genomics, you know that there is no recurrence of mutations, so infrequent. Most mutations are infrequent. So that way, unlike pathogen, for example, in case of COVID, yes, everyone who gets infected by COVID will end up having that virus and its proteins getting presented. But in cancer, from one patient to another, even if both of them have liver cancer, most mutations do not overlap. Some do, but most don't. So that is a challenge, because for you to develop something as a vaccine, you want something which is recurrent across people, so that you know that if you prime the immune system, it will benefit a lot of people from to prevent cancers. But that is not the case. So that's why it has been challenging, because of the lack of recurrence of many of these variants. But nevertheless, people still want to leverage and see, can they prime the immune system against specific, like even today, it is already possible on an individual basis. Like if there is a cancer patient, you can identify using proteomics methods that we use in the lab, what are the antigens being presented on the cell surface of the tumor of that individual. But the only issue is you can't scale that easily, because it is very individualized. Each patient sample has to be processed and analyzed independently, and you can't make it, okay, once you analyze 100 patients, you now know exactly what is the repertoire in the remaining 100. It doesn't work like that. But they are useful in a personalized therapy setting, where you can clearly see which are the antigens being presented, and you can create T cells that recognize those antigens and treat the patients.
Got it. Thank you so much. Yeah, I honestly, I hadn't heard about cancer neoantigens before having a conversation with you. And so it's just so cool. And I think it again speaks to, like you said, even in COVID, we're really harnessing the immune system to prime it and work
Yeah.
it to get ready. And it's really cool that we're able to now start doing this in cancer as well. So I think maybe we can end there. Is there anything else you might have wanted to talk about, or any questions you had for me, or anything as such?
No, so that's it. So like essentially, our lab broader interest is cancer biomarkers and targets, and they could be targets in a conventional sense, where there are these apparently regulated proteins which can be targeted using a small molecular antibody. Or we are also interested in essentially mapping these neoantigens presented by cancer cells, so that they can be leveraged for cancer immunotherapy purposes. So that's the broader interest, and we are doing it in the context of melanoma, breast, lung cancer. And also our interest is to identify, because even within the immune therapy context, it is unclear a priori most of the time who will respond to these therapies and who don't. There are some markers, but they are not necessarily foolproof. So that way they are not, let's say, the best markers that we have. So our lab also investigates what are the biomarkers that we can find that can predict which patients are likely to respond to a given immunotherapy and which who are not likely to respond to a given immunotherapy. So we will continue on those quests. And it was a pleasure talking to you.
Thank you so much, Dr. Harsha. I really appreciate it. This was great. Yeah, and thanks everyone for listening in, and we'll see you next time.